then for each modification, what's the function? And then what's the temporal order that it happens? Okay, so starting at the 5' end of the RNA is capped. So 5' capping, i.e. the addition of the G, the guanosine, and the methylation of that G, because the cap is 7-methyl G. So that's one modification. And that so the modification protects against degradation, right? Because there are very few nucleases that will degrade a 5' phosphoester bond. So then at the other end of the molecule, a modification is 3' polyadenylation. And we talked about the hexanucleotide sequence that you need for the polyadenylation and how that works. And so polyadenylation also protects against degradation. And then both the 5' cap and the 3' poly-A also function in translation. Remember, we have translation factors that bind the cap and help recruitment of the ribosome to the mRNA. And then the third modification would be the paper that you did in section, and we also talked about in lecture, splicing. Splicing happens after transcription, and so the function of splicing is to create diversity, right? Because from a unit of transcription, we can splice in different ways to give different protein products, like big T and little t in the early RNA, or the different capsid proteins from the late RNA, the SV40. So the order, the temporal order of those modifications is exactly the order I gave them to you. The capping happens really quickly within the first few dozen nucleotides of the transcript. Then happens polyadenylation, and then splicing is usually a really slow event relative to the others. All right, any other questions about C? About that question? Any other questions? Yeah. Go over 6E. Okay, the question is 6E. Suppose you are an undergraduate, that's for sure, working in a lab, and you discovered an unusual virus that was captured in a cheese factory. You suspect that the virus might be a retrovirus. So what type of polymerase would you expect the virus to possess if it indeed was a retrovirus? An RNA-dependent RNA polymerase? No. An RNA-dependent DNA polymerase? Yeah, reverse transcriptase. Right? DNA-dependent DNA polymerase? No. No virion-associated polymerase. So it's an RNA-dependent DNA polymerase. Oh, right. I didn't get to the end. That's probably what you were wondering about, right? So suppose you read Professor Baltimore's 1970 paper. I hope you did, because you did it in section. Okay? And in that paper, it describes the characterization of polymerase. And the assay that he used contained, in addition to virions, so the assay, well, this is the assay that Baltimore used. It had virions, and then as a source of the polymerase, the virions contain the polymerase. And then in addition, there's a buffer, then the deoxynucleotide triphosphate, so you have deoxy ATP, deoxy CTP, deoxy GTP. Then almost all polymerases need magnesium. So there's magnesium, in this case magnesium acetate. And DTT is a reductant, dithiothriotol. This is just like detailed stuff, but that was, I think, in the information in section. And then tritiated TTP. So you have radioactively labeled TTP. So that's the assay mix. So if you really have a retrovirus, what amounts of radioactively labeled product would you expect if you incubated this mixture? You incubate for 1 hour at 40 degrees under various conditions. And so the first condition is you delete deoxy ATP from the reaction mixture. Would you get a high or a low amount of product? Because that's what I'm asking you for. High would be greater than 80% of control, low is less than 20%. So if you leave out, we have deoxy ATP, deoxy GTP, deoxy CTP, the deoxynucleotides plus the thymosin triphosphate. So if you leave out deoxy ATP, what would that do to affect how much product you make? Yeah, you would make a low amount because you need all 4 nucleotides for the polymerase to make a product, right? In the absence of A, you know, if you came to a the complementary base to an A, which would be no, it's an RNA. The template's RNA, right? Because this is made, the polymerase is reverse transcriptase, but the template is plus single-stranded RNA. So there'd be a U in the RNA that would go to A, right? So when you came to the first U, you wouldn't have any A to incorporate. So it would terminate. Right. So you'd have a very low amount of product if you left out deoxy ATP. Then the next part of the question is if you replace the deoxynucleotides, so deoxy ATP, CTP, GTP, and the TTP, if you replace those nucleotides with the ribonucleotides, so ATP, CTP, GTP, and UTP, what would happen? Well, it's a DNA polymerase. It doesn't make RNA, so you wouldn't get a product. It doesn't incorporate ribonucleotides, it incorporates deoxyribonucleotides. It's not an RNA polymerase, it's a DNA polymerase. Okay. Are we comfortable with that question? Any other questions? Next. Yeah. How do transactivators work? Do you have a specific one in mind?
Just had it under because it wasn't a transaction. So that question.
I didn't ask you for an example. Where is this? 8A to B. Okay, so how does a transactivator work? So activation, right? So it's a protein that's going to work in trans to increase transcription, to activate transcription. So what are those proteins that we consider? protein a day early, and that's targeted back, it's a transcription factor that you need to activate transcription of another set of genes. In this case, the beta gene. So that would be one example of a transactivator. Another example would be in HIV, TAT. Because TAT acts to recruit the cyclin-dependent protein kinase, the cyclin, to increase polymerase activity, right? But it acts in trans. So your question, would an enhancer binding protein qualify? It certainly activates transcription, but I'd say it wouldn't qualify in this sense in the way that we've defined it, because we're asking for viral gene products that activate the trans to increase transcription. Like the herpes example, the HIV example I gave you. In the enhancer binding proteins that we've discussed, none of them are viral gene products, right? So that wouldn't work. You okay with question 8? Yeah.
What do you mean when you say works in trans?
Well, trans, I guess, would be in contrast to cis, within, right? So it's a protein that you've expressed that's then going to target and act at a different element. Typically, a cis element is how the language goes, right? Because the element is within the same piece of genetic information whose expression you're modulating. Do you talk about cis and trans in genetics? Not too much, it sounds like. And it's kind of an important concept, actually. So cis elements are, how we've discussed them in terms of genetic regulation and virology, are elements within the piece of viral genetic information whose expression we're modulating. And we're modulating that expression by virtue of a protein binding in trans at that element. Although today we talked about two cis elements that are not viral. Right? For example, all of the interferon-regulated genes, of which there are several, if they're regulated by alpha interferons or beta, those genes all have an ISRE element. So that's a cis element that's present in these different genes, to which the transcription factors, in this case the stats, right? So the stats would be the transacting factors that act at that cis element. But that's all cellular, it's not viral.
Could you go over 7B, the incubation period for AIDS, as the clinical studies of transfusion cases in the U.S. and the average age of diagnosis of AIDS?
Yeah, so Dr. Sears went over this. He had a slide on it, actually. So I didn't go over it. In the sense of the age, right? Because usually after the infection, the individual would be HIV positive relatively quickly, right? But development of the syndrome, AIDS, is less quick, right? And so transfusion cases are no different though than the slide that he gave. And so it's a window of time. Transfusion cases sometimes are viewed to be a good estimate because there's fewer other complications so that period of time is 7 to 9 to 10 years, which is kind of, he's got a slide that he gave to you in his lecture, and it's on the website that has that information. So anything in that window, 7 to 10 years, if you find, if I happen to ask that question. Because the answer that came out of different studies is slightly different. That's why it's a window.
For that same question, I think.
What is the question?
Oh, the average age of.
No, I mean on the
Oh, seven.
7,
7B.
7B, right?
The average age of diagnosis is like 8.
Oh, okay, yeah, you did ask. So it's in the mid-30s to 40? What else did I ask?
For that same question, the leading
Yeah.
cause of death for females in the age group, I know males is accidents and cardiac disease, what was the female?
No, that's not right for males.
Is that including HIV then,
Yeah.
during that time period?
And that's why I gave you,
Yeah.
I showed you the slide, right? And, so in that age group, leading cause of death for males is AIDS and accidents. Leading cause of death for females is cancer. And for females, the leading, the causes of death more or less have been flat, steady over several years. Those lines didn't really change on the window that I gave you from CDC.
This is from a different year. I mean, right now, AIDS is the leading cause. It's from, like, what year are these statistics from?
These would have been about 10 years ago, but in terms of the percent of people that have survived or not, that doesn't substantially change. People are living longer because of the therapies that are available. That's why the curve started to go down. The therapies, but the other part that I gave you, because I happened to ask that question, I am thinking about education. People would say education is an important point too. And I showed you some slides that I collected if you were in class that day.
Could we go through parts F through H of question 7?
F through H,
Yeah, I
and
think
that
probably
same
that's-
question?
Yeah, question 7, H 1.
Well, this was kind of asked F earlier, correlation of the biophenotype of HIV disease progression has led to M-trophic and T-trophic designations for virus. Two characteristics of T-trophic relative to M-trophic virus are, so one of the points that we talked about earlier is the chemokine receptor that's used, which is usually the alpha receptor at late stages, CXCR4. This ability to induce syncytial formation in cells differs. Essentially, the fusion of an infected cell with an immune cell because of the fusogenic activity. T-trophic viruses replicate in T cells pretty efficiently and are seen with accelerated disease progression. Synthesis of HIV message is catalyzed by the cellular polymerase, right? So you
just,
don't get HIV mRNA synthesized until you've made the provirus,
Right, so
which
it's
has
just
to be
cellular
integrated into
RNA.
chromatin. And once you're to that stage, it's cellular machinery that transcribes. You're using the viral cis elements, wherever that question was earlier. You're using the viral promoters and enhancers cellular RNA polymerase II and general factors that make the message. So that's G. Then H, set of criteria used to assess the role of an agent, like a virus, in a given disease. So these are the so-called Koch's postulates that I listed. So it's the criterion that logically have to be fulfilled to establish a cause and effect relationship between an agent. And it wasn't worked out with HIV or AIDS, it was worked out, decades earlier in the study of anthrax, but, the concept is the same, to establish cause and effect relationship.
So that question 7F, when you mentioned confusion activity.
Seven, which?
7F, is characteristics of T trophic.
Characteristics of T-trophic relative to M-trophic viruses.
So T trophic viruses are more likely to have fusion activity.
Right. So they have sensational inducing activity.
The entropic ones, some of them?
They less so.
Oh, less so, okay.
Right. Than the chemokine receptor that would be utilized. And then associated with accelerated disease progression. That was kind of a meaty question, wasn't it? A lot of stuff to remember. Right. I try to give you some questions you have to think, like those protein synthesis inhibitor ones. You have to synthesize information and put it together. Then other of the questions, that's just like, can you remember the information effectively? Any other questions?
Can we go over GG and HH on problem 4?
Yeah, I'll probably have some of these kind of questions on the final. I mean, we had them on the midterms, right? But bring a pencil, because we'll probably use a scan from. Okay. GG and HH. GG, mechanisms by which a cellular homolog of a viral oncogene, so the C-onc. What are mechanisms by which the C-onc may be activated to become transforming genes in animal model systems? And those mechanisms include chromosomal rearrangement by reciprocal translocation. Well, that's definitely a mechanism. We talked about that, right? Like the Abel oncogene would be a good example. The translocation that gives BCR Abel. Point mutation leading to an amino acid substitution. Would that work? What was the example? No, point mutation. That leads to an amino acid substitution. Yeah, but what's the oncogene? RAS. There's a table on the handout, remember? So point mutations in RAS at certain positions in CRAS have been associated with some human cancers. And I gave you a table of that, with the hot spots in RAS. So translocations, yes. Amino acid substitutions, yes. Promoter enhancer insertion. So the answer is yes for that too. And the example I gave you is avian retrovirus that's oncogenic, even though there's no obvious oncogene in the virus. And so what happens is the insertion occurs in the position to drive up the expression of the c-Myc gene in the avian body by providing viral promoter. So the answer would be all of the above for the GG.
So does that mean CRAS would, in this question, turn on the C-onc or homologous? Are they?
No, C-RAS is the C-onc.
Okay.
I mean, RAS is an oncogene, right? That came actually from the murine sarcomavirus. There are actually two RASes. Harvey, you know, the second one, can't remember that. But they came from mouse viruses. And so the murine, the mouse retrovirus, which is a sarcomavirus, had an oncogene. And it was designated RAS. Just like the avian Rous sarcomavirus had an oncogene that was designated SARC. It came from avian sarcoma, but Peyton Rous is the sarcoma virus. So the oncogenes were identified in the virus, and then again, it was found that these different retroviruses that have different oncogenes, cells have a homolog of that oncogene, or those different oncogenes. That's actually the Nobel Prize. That's the basis of the Nobel Prize to Bishop and Farmas in San Francisco. The whole idea of cellular foot oncogenes, right? So in the context of the virus, we touched on it a little bit, but I didn't really dwell on it this year. I mean, the viral V-onc and the C-onc, they aren't exactly the same, but they're really, really similar. The viral gene has accumulated mutations usually that give rise to an enhanced activity. But they're basically the same gene. All right. So what was the other one? GG and SOGRAS? HH. The regulatory protein TAT functions to enhance LTR-specific transcription of HIV by, well, we kind of talked about this. I mean, it's transactivated, right? So binding to the TAR DNA structure to recruit DNA polymerase. No, that's not right. I mean, TAT is the HIV protein, but it binds to an RNA structure. It binds to TAR. So binding TAR RNA structure to recruit cyclin D1 and the cyclin-dependent kinase CDK9. That's what we did talk about, and the answer is yes for that one. Binding to the RRE RNA structure to enhance splicing. No, that's not true. I mean, that's what binds to RRE. Rev, right? But it doesn't, it affects transport. And if you move the RNA out of the nucleus, then it's not in the nucleus to be spliced. Yeah, so Rev is RRE. 4, back, what is TAT doing? By increasing the phosphorylation of RNA polymerase and enhancing transcriptional elongation. Well, that's true. I mean, the study of TAT and TAR is actually what, provided understanding as to how elongation increases for RNA polymerase 2. And how the C-terminal tail, Pol2, gets phosphorylated, which pushes you into the elongation mode. Right. Okay. Any other questions? Next question.
11 or 12B, the last question regarding
this is the proof of concept. So if Able, if you had the chromosome translocation, so following that translocation, then the C-Able gene ends up being expressed as a fusion with BCR. Right? So you end up getting a different protein, BCR-Able, in the translocation, which is seen with chronic neurological CML.
Is that the
So,
virus? Is CML the name of the virus?
no, there's no virus. This is a human cancer.
Okay.
But the oncogene was originally identified as the cellular C-onc homolog of some viral oncogene. You know, the viral oncogene happens to be, well, I guess I did ask a question, homolog of the oncoprotein encoded by, well, it'd be Abelson murine leukemia virus, right? But what Gleevec is a drug that was discovered that inhibited that tyrosine kinase in the fusion form, right? But not the normal form. So, you know, it's a proof of principle idea that if you can identify the principal component that's responsible for the proliferation, enhanced proliferation of cancer, then you might have a chance of getting a drug against that component. And so that was done in Gleevec and this tyrosine kinase activity, because Gleevec inhibits the BCR-Able form of the tyrosine kinase.
And so Gleevec actually doesn't treat a viral cancer, it actually treats the cellular CML.
What's the human disease? But the human disease has nothing to do with the virus.
Yeah.
It has to do with the translocation of genetic information from one chromosome to another by, you know, whatever causes translocations. Radiation can cause translocations. And as a consequence of that translocation, the expression of the cellular component was altered. And that cellular component happened to be the C-Able gene, because it becomes fused with PCR.
So as far as just filling this one part, it would, you would just put the first blank C-Able, because it's asking for what the homolog of the oncoprotein is itself. So the first blank C-Able MLB.
So the drug STI 1571, which is known as Gleevec, is directed against the blank enzyme. Well, the enzyme best answer would be tyrosine kinase, because Able is a tyrosine kinase. But you could say Able kinase. Because I asked, the drug is directed against what enzyme? Right? So the enzyme is a tyrosine kinase. It's a C-Able tyrosine kinase. And that enzyme is a homolog of the oncoprotein encoded by what virus? And the virus was Abelson murine leukemia virus.
For under the test, when you have to put in the name of the virus, is it okay to use abbreviations like HIV, MLB, or you want to write down the virus?
It's always safest to write out the name of the virus, because there are some abbreviations that are the same, sadly. And what would those be if we talked about, like RSV if you know actually? Because RSV, what does RSV mean to you? Probably means Ross sarcomavirus, mostly, right? Because that's what we mostly talked about in that context. But I also talked about respiratory syncytial virus briefly, right? And that virus is RSV. So, you know, look, abbreviations are fine in science as long whose activity may be altered in cells infected with RNA viruses include TBP, TF2D? No, definitely not, because that's not a protein synthesis factor. That's a transcription factor. It's, TBP is taught a binding protein, speaking of abbreviations. And TF2D, TBP is sometimes called TF2D or part of 2D. And we discussed that factor when we discussed SV40, because that's when we discussed Box that was identified the positions the start of transcription in terms of where transcription starts. And that's affected by that factor. So it's definitely not a translation factor, protein synthesis factor, it's a transcription factor. Okay, 2, P220 EIF4G. So the answer is yes to that, because that's the factor that's inactivated in polio-infected cells that leads to the inhibition of CAP-dependent translation. Right? It's kind of the flu question early on when we were talking about mixed infection and polio. So yes for 2. EIF2 alpha. Definitely yes for that. We discussed EIF2 alpha when we discussed reovirus as the first virus in infections where you get activation of PKR and phosphorylation of EIF2. And we discussed EIF2 today, actually, in the context of the substrate for PKR and the inhibition of virus growth in an interferon-treated cell and has induced levels of PKR. So the answer is yes for 3. 4 is 1 and 2, so that can't be right because 1 is 5 is 2 and 3, so that's yes. So the answer to G is 5, 2 and 3. Any other questions? Okay, I have some questions for you. So, any viruses that maybe that you would have liked to talk about in class that we didn't cover? Because there are plenty of viruses, and I cover some of the same classic ones every year, but some years I change it around a little, depending upon sort of what seems to be a current issue. I guess we're okay.
Not like a specific virus, but I wish we could like another full day on just like what was going on currently, like how we talked about Zika virus and
Yeah.
that outbreak, just like maybe one more day.
Yeah, I was kind of hoping to do that, because I have a cell with Ebola, for example. And when Dr. Sears gave a couple of lectures, I was actually in Germany for a couple of lectures at Marburg, where the Marburg agent came out of the same family as Ebola, but it's subsequently developed. So while Zika is in the news this spring, last year it was Ebola.
Yeah.
Right. Yeah. Yeah, I like that too. I mean, it's kind of a. Anything else? The other thing I was thinking of doing in the handouts, rather than giving them at each lecture, is I'll just, have them made into a booklet and you have to buy it versus each lecture. Hmm?
I think that's a good idea. Because like you can get, because I'm not very good at keeping track of mine. So like I lost all the handouts and I have to go look at them online again.
Right. But
I'm
that's
saying-
kind of why I
Yeah.
finally decided, I'll just like bullet and copy them for you and bring them to class. So you way to go. But, you know, you probably figured out that many of them are in color on the website, right? So if you have a color printer, you can get a much nicer handout for the cells that I've made. Whereas, you know, we can't afford to give you color handouts.
For the retroviruses, HIV has its protease in a different gene than all the other ones, right?
Yep.
Like it's typically in Paul, or is typically in GAG and.
Which virus are you talking about?
I'm talking about all the retroviruses. and
Okay,
then-
whoa, stop right there. So if you're talking about all of the retroviruses except,
Yes, except HIV.
okay, so it's like I taught you at the beginning. It's like Ross sarcoma virus. The protease is in the GAG gene, okay? For the bulk of the retroviruses. The exception, the except to that is that the protease is in the Paul gene for HIV. So that should be on the handout, right? Because PR is in one spot or the other.
I copied the wrong class. Okay.
But it's in the hand, you got your HIV, I should have brought the hand, speaking of the pound. Anyway, if you look at the handout, and I have a schematic that has the genes of different viruses, you see PRs in one place or the other, and it's in the Paul gene for HIV. So I guess what that really means is that you can't make any mature protein until you had the translation with the frame shift to make the GAG-Paul fusion. You have to make the protease.
Does GAG need to be cut with the protease in order to be functioning?
Oh, yeah, for sure.
Okay.
Right, because we have these different mature proteins that GAG is simply a precursor, right? Paul is also a precursor. We call it GAG gene or Paul gene, but the GAG is actually a polyprotein precursor that for the bulk of these viruses, like Ross sarcoma virus, that was our model virus, because that's really where it was figured out. You autoproteolytically cleave down that GAG precursor to make these various internal, capsid, nucleocapsid proteins. Right. It'd be conceptually like polio, where we make in the case of polio or flaviviruses, you make a very, very large polyprotein precursor that has a couple of protease domains that self-cleave down. For HIV, you only have one protease. It's different in that sense. HIV has one protease domain, but where it is depends on the virus. It's in GAG for all the guys except HIV. For HIV, it's in Paul. And to finish the train of thought, so Paul is a precursor too, in a sense that there are two different polymerase subunits. You have the reverse transcriptase polymerase, you have alpha and beta subunit, and you cleave out RNase H and integrase, right? Those are Paul gene products, but they're not polymerase. They're the integrase and the nuclease that you use to make the protein.
So with the twitching of the polymerase to make the GAG-Paul fusion,
Twitching with the polymerase.
well, it's not even right, twitching.
Yeah, I haven't
Frame shift.
heard.
Anyway, frame shift, but, so Paul the GAG-Paul fusion, like is Paul itself GAG-Paul? Not like, it doesn't
You
break off.
can't have
does it?
Paul without GAG.
Yeah. So like, is it two different proteins, GAG and Paul, or is it GAG and then GAG-Paul?
It's two different genes. One gene is GAG and one gene is Paul. The GAG gene is expressed from the full transcript, right? That was made by the cell polymerase from the integrated probiotics, right? No splicing, full-length transcript, it's out into the cytoplasm. It has an AUG, you initiate translation of this big, long, open reading frame, so we have a full-length transcript. But it's gonna have an open reading frame. It gives the gag precursor. And then there's a different reading frame for Paul. But we translate and we make this gag protein, and within this we have protease domain that gives us cleavage down. That happens 95% of the time. 5% or so, we would have the frame shift that allows us to get into the Paul open reading frame without terminating, so the frame shift happens obviously before the termination codon. So in that situation, you don't make gag, but you make a precursor that's the gag Paul precursor. So you can't translate this without having translated gag ahead of time. Right? And then this gets cleaved down by the protease to make the Paul. You know, whether you get anything out of gag, I have no idea. It's probably insignificant because you're making enough gag anyway. Got it?
Yeah, no, I'm not gonna ask another question.
Oh, well, he had a question.
For poliovirus, when it's, I think replicating its genome, why does it circularize its RNA?
Well, it circularizes its RNA because proteins are being bound to that RNA at both the 3' and the 5' ends, and then interact with each other. All right, so the actual polymerase is going to transcribe from the 3' toward the 5' end, right? But the RNA structure and the initial recruitments are at the 5' end. And then, as among those proteins, and you have this on your handout, are the VPG protein that, so it's a viral protein that gets uridylinated, so it gets a couple of U's added to it covalently. And then those U's are complementary to the A's at the 3' end, so that, you know, the other end of the RNA had to be in proximity for that interaction to happen to allow for the priming. The
Okay.
initial binding is in RNA structures at the 5' end.
So it's just to bring everything closer together.
That'd be one way to look at it.
Thank you.
But it's through specific protein-protein interactions. Okay, any other questions?
Yeah, I just had one of the multiple choice questions I forgot to ask the first time around. Well, two of them actually. The first one is the X, like the very last one. There's two X's, but this is the one at the very end of problem 4.
X. Very last X?
Yeah. That's the-
After KK.
After KK.
What am I doing to help? The synthesis of viral RNA transcripts. This is the last one. The synthesis of those viral RNA transcripts takes place in the cytoplasm of cells infected with human viruses that cause AIDS.
No.
No, because the viral RNA transcripts are made by the cell polymerase. Well, too, and that's nuclear, right? We talked about that earlier. So, no for AIDS.
suspicious when it's not, when the answer is that has multiple choices.
Oh,
So, I just
you
wanted
mean
to
you
check.
profiled me?
Yeah.
I have to remember that.
And then the other one, I just lost track of it, sorry.
I mean, these kind of questions, I guess, allow one to ask a lot of information in one question, right? And there's multiple kinds of answers.
I mean, I'm just like generally suspicious of that kind. It's not like your class specifically. I feel like in most science classes I take, if there's like a all of the above or like multiple of the above, it's like more often than not
Uh,
correct answer.
Yeah, I mean, there was nothing premeditated in my mind in terms of how I framed the questions. Of making them multiple as opposed to single.
I can't find the other one.
For today's lecture, we went over interferons. Can you go over the kinase again?
Okay, so in the context of today's lecture, how does PKR come into play? So PKR is a gene that's induced by interferon. So when a cell is treated with interferon, in that JAK-STAT signaling happens to induce transcription, one of the many genes that's induced by interferons is PKR, the protein kinase that's regulated by RNA. So in the interferon treated cell, when you interferon treat, then you induce PKR. So you make a lot of this protein, whereas with no interferon, there's much less PKR, just a little protein. But when you interferon treat, you induce the gene because of JAK-STAT signaling, and the gene has an IS3 element. You make the protein, but the protein isn't active. It has to be activated. And it's, as we just said, it's protein kinase R. The R is because it's activated by RNA, double-stranded RNA. So you can get double-stranded RNA made in the infected cell by replicated intermediates and the like that we've discussed. So when you have double-stranded RNA, i.e. virus infection, PKR autophosphorylates itself. And this is on the handout. So when it gets phosphorylated and it phosphorylates itself, then this is active. The unphosphorylated form is inactive. So when PKR is activated, then what it phosphorylates is EIF2. So when you have phospho-PKR, then that can phosphorylate EIF2 to give EIF2 the alpha subunit, it happens to be serine 51. So when EIF2 is phosphorylated, then this factor is inactivated, doesn't work in protein synthesis initiation. So the interferon treatment induces PKR. The viral infection provides an RNA that activates PKR. When PKR is activated, it phosphorylates the initiation factor to inactivate it. And then you have an inhibition of protein synthesis in those treated infected cells. And I showed you a slide illustrating the inhibition of rhabdovirus protein synthesis, right?
I found my question. It was just once again multiple
Hang on,
choice.
so you got it?
Oh,
Is that
sorry.
okay? Okay.
yeah, sorry. Do you want me to, can I change the subject now, or are we done?
No, she's finished.
Okay, cool. Yeah, question D on the multiple choice section. Guanine nucleotide binding proteins include.
Okay. D. Guanine nucleotide binding proteins include RNA polymerase of reovirus?
I doubt it. I don't know though.
Well, yeah. I mean, the polymerase, and we talked about it right here, right? Polymerases need the 4 nucleotide triphosphates, either ribonucleotides if they're an RNA polymerase, or deoxynucleotides if they're DNA polymerase. So, reovirus RNA polymerase would certainly bind a guanine nucleotide.
By binding,
GTP.
by binding the guanine nucleotide, are you talking about adding to like a growing strand?
Well, it's a substrate, right? The enzyme has to bind the substrate. The polymerase is the enzyme. GTP is one of the substrates, like ATP, UTP, and CTP, right? So the answer, so yes for 1. Initiation factor EIF2. Well, the answer is yes for that. Because EIF2 binds GTP in the initiator tRNA. Remember when we talked about cap-dependent translation initiation?
I don't remember that.
You remember? Anyone? Yeah, so we did. When a scanning model of translation initiation. So the answer is yes for 2. Harvey murine sarcomavirus oncogene HRAS. So the answer is yes for that, because RAS is a GTPase, remember? So the answer is all of the above. I guess your predicted answer.
Oh yeah.
But do you understand the question?
Yeah, I understand the question now. I definitely need to go over some of the answer choices. Like, really just the initiation factor. I don't really know what that is still. I know we've been talking about it.
I mean, I try not to pull really obscure factors out of the bag, but EIF2 we've talked about, and EIF4, is pretty detailed. Because they're, kind of the heart of mechanisms.
Are those the ones did we talk about that today? Wait, is that like an interferon? Is that what we talked about today?
No. Well, EIF2 came up in the discussion of interferon and PKR today, but EIF2 came up in the discussion of translational control and scanning, and that actually went back to reovirus, a lot of their basic work with DSP.
Okay.
Remember AUGs and what makes a good AUG or a less good AUG?
Talk about like partial conditions +4.
Yeah, I mean, it was that same lecture that I talked about some of this stuff.
Wait, so the IF2 is involved with that? Like looking at the.
Oh, not
Oh.
that part,
Okay.
but secondary structure is involved because that's double strandedness, which can activate PKR and phosphorylated too. And then, like an RNA polymerase, that's genetics, right?
Yeah.
Got that a long time ago.
Yeah.
Got it from me again. Okay. Anyone come to the lecture today at 11? In this room, actually? Did you?
Oh wait, was that the one? Who was doing that?
Who gave the lecture?
Yeah.
Carol Greider.
Oh, I went.
She was did you come? Good.
Yeah, I'm in the stem cell class. That's like,
It was a great talk.
yeah, liked it.
She was an undergraduate here a couple of years ago. Maybe, I forget, 20 years ago or something. of genes on the chromosome. But in addition to all the genes, of course, that you think about with chromosomes, there's a centromere that's really important. But there are the ends of the chromosomes that are pretty important. And so the ends are the telomeres and the telomerase that basically keeps the end in position. Because if you lose the end of the chromosome, you know, it's essentially you're losing the buffer, and then you would lose the protection of the genes that are on that chromosome. So she was one of the two major people who worked out those mechanisms. And for that, got the Nobel Prize in medicine. Half dozen years ago, roughly. When was it? You were at the lecture.
Oh, I don't remember.
Yeah. I just kind of remember as an undergraduate running around the lab. Not my lab, but next door lab. Okay, a couple more questions, then I'm gonna go to another meeting.
Do viruses have the end replication problem?
Do viruses have the telomer? Well, no RNA viruses have that issue, because these are DNA.
We don't have the same shoots as to the big lives, so like the logging in, you can't put a primer on, you can't make the.
No, RNA viruses use RNA polymerases. So you're saying, can the RNA polymerase go all the way to the end of the template?
On the lighting strand.
No, there's only one strand that we're making here. Right. You know, RNA is very different in another way that hopefully you remember from class. None of these polymerases are perfect. They can make a mistake. And for RNA polymerases, it's 1 out of 1,000 to 10,000, usually closer to 10,000 nucleotides. You'll have a misincorporation. But once that happens, you're stuck with it, right? Because there's no proofreading to correct errors. So essentially, as you go through replications, this contributes to what's called quasi-species populations for an RNA virus.
What about the DNA viruses?
Well, that's, yeah, what I was kind of thinking. You know, I haven't really considered it or thought of it. It's a good question. In terms of how the DNA polymerase gets to the end, because you don't have these repeated sequences other than, say, herpes that we talked about. It's a good question. I don't know the answer to the question. Should have asked Grider.
I was in section.
Oh, you were in section. Good. I'm glad you went to section. That was a good question. You didn't ask it.
Oh, I didn't hear.
No, I was a little late coming over here because I was talking with her actually. Didn't occur to me to ask. Should have talked with her after, then I could ask your question. Okay, 2 more questions. Right.
I have one basic one. It's what does the GP37 RSV do? The protein, the glycoprotein 37 in Rous sarcoma virus.
Well, the retroviruses all have 2 glycoproteins. One of the glycoproteins is the attachment glycoprotein, right? And one of the glycoproteins has the fusion activity. Is a conceptual way to look at it. And so that wouldn't much matter which the virus is. And so the larger of the 2 glycoproteins would interact with the receptor and the host, depending upon the virus, whatever that receptor is, right? And the smaller of the 2 is the one that usually has the activated fusion activity. So on your, you have the HIV handout, because that would be on. So on the HIV handout, I showed put it in a little more detail, because it actually turns out to be one of the newer drug targets that we mentioned but didn't really dwell on. But again, it's the larger glycoprotein. In this case, it's the GP120, right?
Yeah.
Or the SU glycoprotein, the surface, that's interacting with the cell receptor. But that would be conceptually the situation for osteochondromas too.
Okay.
Then the other glycoprotein, following that initial binding, leads to a conformational change, so you activate the fusogenic activity, so you can mediate fusion between the envelope of the virus and the plasma membrane of the cell, releasing the nucleocapsid into the cell.
Okay.
Right. So I gave that a little more detail in the HIV handout, but still you have two glycoproteins, which was what your question is. I gave it a little more detail because in that entry window are where drug targets are. New drug targets. And new drugs to either impair the interaction with the receptor or to impair the fusion activity of the protein that has the fusogenic activity. Those are both targets. Right.
Yeah. Okay.
Can you talk about the significance of MAP kinase? Or like the regulatory pathway?
MAP kinase. I didn't really talk about MAP kinase in class, did I? When did I talk about MAP kinase?
I'm not sure. I think you said that it was important because it prevented translation.
I mean, there are lots of MAP and ERK kinases, but I can't actually think in what context. I can't place where I. Does anyone know where I talked about MAP kinase? I don't think I talked about MAP kinase in this class. You know, some of those, I talked about cyclin-dependent kinases, like CDK9 and recruitment with cyclin by the cat-tar interaction, which leads to the phosphorylation of the RNA polymerase C-terminal tail. So I definitely talked about that. And I'm raising it because that would be kind of a different sort of kinase. But I don't think we talked about MAP kinases. That doesn't mean they can't be important. They're certainly important as signaling molecules, and a lot of signaling pathways are altered in infected cells. Okay, any other questions?
For the, will you be asking about the specific mechanism for retrovirus replication, or should we just know the general mechanism?
Well, I took the time to make a handout for you, so that should signal that I think it's kind of important, right? I also made the handout in detail because it's kind of complicated, right? So you should know the basics of the mechanism. Okay. Now, I don't know, I have to decide between now and Monday, right? I have a couple of days to get this exam ready.
And then another question?
But you've given me food for thought.
Why was the HIV genome larger than RSV genome, given that HIV lacks the extra oncogene?
Oh, but it has a bunch of other genes.
Oh, okay, so it's really important.
Yeah, a bunch of other genes. The very complex splicing of those genes. Right? Got it? Got it.
What did you talk about the leaks, Abby?
Well, it would have come up with reovirus, but it's come up with other viruses as well, because any mRNA where you have two sets of translational punctuation points, right, that are used. What allows that to happen is leaky scanning. And so we have the 5' cap, 3' poly A. So we have an open reading frame that begins with AUG. As translation starts and it terminates at some stop codon, one of the three stop codons. So that's an open reading frame, right? And then say there's so what makes this a good AUG or a bad AUG? So this is what we were talking about a little bit ago. Depends on the flanking nucleotides. And -3, so if the A is +1, going back 3 positions, this position and this position. And if -3 and +4 are purines, so that means an A or a G, that makes that a very efficient AUG. It works really well. Because when the 40S subunit binds at the 5' end of the cap, 40S subunit, then that subunit has to scan till it comes to an AUG. And if it's a strong AUG, it'll stop almost completely to bind the 60S subunit and start translation. But if it has a pyrimidine in either -3 or +4 or both positions, then that makes the AUG weak, less efficient. And some ribosomes would bypass it. And so if you have an AUG in a different reading frame, that uses a different stop sign. So let's, this frame. How you get the ribosome to that AUG to start translation, it has to be a leaky scan to bypass the first one. And that will happen, when you have pyrimidines in those two positions, just empirically that's been worked out. Then you don't want to have double-stranded RNA, because double-stranded RNA makes the scanning less efficient. There's a helicase that can destroy that RNA structure, but still more structure, less efficiency generally. Because of the scanning effect on scanning ribosomes, also because it presumably could activate PKR. So that's where leaky scanning. So it was kind of real bars lecture that we talked about translation like this. And then, but SB40, two of the